Thank you very much for the introduction. And I did slightly adjust my title, but you're in the right place if you've come to listen to snap bean disease management updates. First off, I just wanted to thank the Midwest food producers and Del Monte and our other partners in this work. This was the product of the third year of our funding cycle for field trialing to look at specific fungicides and their timings for disease control in snap beans. And we very much appreciate that funding. Particularly in this final year, we had a lot of hiccups and uncertainties with respect to getting our trials out due to the challenges with COVID-19. And so we were able to get our field trials out. Things were a little bit late, and I'll explain how that has impacted our outcomes. But again, I just wanted to express my appreciation. And secondly, I wanted to acknowledge the co-author here, Stephen Jordan. Stephen set up these trials and was able to work with our students, all graduate students that were employed this summer and could assist us in the field and certainly in data collection and analysis and summary as well. So certainly want to acknowledge my co-author on this piece. So to start off this root rot and damping off diseases update, again, this is the final year, but we will have plans to continue with likely a perennial basis in continuing to investigate seed and at planting treatments for disease control in snap beans. In particular, we really used and abused a particular hotspot at the Hancock Ag Research Station. It had great disease to the point where it was too much. And so some of our capacity to discern treatments was hampered. And sometimes that's a good problem to have, but in this year, it certainly did influence our outcomes. So we'll find a new location there, and we've got great interest. We have some treatments that seem to really stand out, and we want to trial them and demonstrate their effectiveness in combination with a whole program. So there'll be more from this ongoing work in the future, but this is the final year of the funded project. And here, again, we're looking at seed applied treatments as well as in furrow or in line at planting fungicides for the control of root rot and damping off diseases. And these diseases are many, although this year, the one thing we did was we brought infected or diseased plants into to the lab or to the diagnostic clinic to make some determinations on pathogens. In past years, we were ascertaining or presuming what we had present based on the effectiveness of particular fungicides and their expected control attributes. But this year we did bring in plants and confirm. And this was a later season planting, and I'll get to the details of the trial. But we primarily saw Fusarium, probably one or two species. We didn't speciate. We didn't take this through to sequencing to determine species. But we had Fusaria and we had Pythium. And this certainly revealed itself in some of our disease management outcomes. But this group of pathogens and their diseases include Rhizoctonia solani. Typically that one is problematic when it is wetter and cooler. We certainly had moisture in the field assisted by irrigation, but it was warmer by the time we planted, and that's why we certainly saw more Fusaria. Among the oomycetes, or the water mold pathogens, we did not see Aphanomyces euteiches. We did see Pythium species present in some of the root and lower stem tissues of plants from our study. But these diseases affect bean crops and other crops annually. They reduce stand by affecting the root systems directly, and therefore some plants never make it out of the ground. Others will affect at the lower stem, and the plant will dampen off or fall over. And again, the pressures which promote most of this suite of pathogens is cool and wet, or cool and warm. So other management or the cultural practices which lead to management of these diseases include varietal resistance. Certainly that's at the basis of all strong disease management programs. Crop rotation out of the susceptible legume is a really important one. Again, we demonstrate that in our field trials that took us down to fairly low yields and canopy coverage this year. Avoid planting during times when the soil remains consistently wet. Certainly that condition, along with limited airflow in the crop, will promote these root rot and damping off diseases. And seed applied in that plant fungicides can certainly help out in reducing losses. So this past year's trial was at Hancock, as I mentioned. We worked on the Huntington variety as well as high style. We planted on the 12th of June, and this was later than would have been optimum for our trial goals. But we were not quite sure what we could do when we got from March to April to May. And finally, when plans were made at the college level that we could do some of this work, we were into the month of June to get things rolling once we had our resources in place. Fungicides were applied as drenches in row over the top of covered seed for our what I refer to as inline or in furrow. And then the seed treatments were applied and dried on the seed the morning of planting. We did that by hand. The data we collected included emergence. So that was sort of a percent emergence. We collected foliar and root symptoms and had our diagnostic outcomes. We also recorded canopy cover this year, in part because it was so low. We wanted to look at that element as it seemed influential or seemed important. And then yield. We hand harvested the crop on the 24th of August. All right, so the treatments we looked at this year, and this is a treatment list that is the product of lots of communication with industry. Folks were wanting to look at particular some of these seed treatments that went in, but I'll start first with the in furrow since that is at the top of our list. And so we had our non-treated control, of course, to compare. We had Ridimol Gold, Ridimol plus Quadris, so that is the methanoxam for the water molds, and then azoxystrobin. We had Quadris alone, Velum Prime by itself. This is fluopyram. And then we had Serenade in furrow and double nickel. Both of those are Bacillus species. For our seed treatments, we looked at Evergoal Energy. And this is one of these newer, relatively newer combinations. And this one has the phenylamide or the omycete material metalaxyl. It also has a group 3 and a group 7 fungicide. So the 3 is prothioconazole, which is also proline or proline. And then it has penflufen as the group 7 or the SDHI in that mix. The next seed treatment we looked at is Vitaflow. This one, I think primarily has registration out of Canada, but there was interest in this. It has Thiram, which is a base protectant or a multi-site. And then Carbothian is the group 7 or the SDHI here. And then we looked at Ridimol Gold as a seed treatment. We were curious to see how that compared to the in-furrow treatment in terms of placement. And Velum Prime as a seed treatment went out. And Saltro, which is pitaflumetophen, and that's a newer Syngenta material, also referred to as adipidin. All right. So our outcomes here. What you'll see first off, we did have very high disease pressure. So our yields, and this is in pounds per plot, quite a low yield. I know when we were looking at these, I thought maybe I had the tags wrong and this was tons per acre, but no, it was pounds per acre or pounds per plot size. So it is very low, and our certainly our canopy cover was extremely low as well. I'm not going to show those data here today. There were no significant differences, but they were quite low. But we did see were some trends that I think are interesting, and we can still take away some learning from this experience. So for high style, which is represented by the orange bars, and the bars are representing the yield. So the taller the bar, the more yield. Going along with the orange bar for yield with high style is the lighter orange or yellow golden colored dot above it. And these are emergence ratings. So that's percent emergence for high style. And then just to situate you in this graphic, the blue bars represent yield for Huntington, and the gray dots represent emergence percentages for Huntington. All right, so when you look across this this graph, and I think folks can probably see my arrow now, I'll use that as a laser pointer. Our non-treated control is on the far left here. So taking just a quick glance across for Huntington, Most of our treatments did better than the non-treated control, although there were a few below, certainly. But those that performed best, and that's the way to interpret these data here, those that performed best for Huntington did include the Ridimil and Quadris. In furrow, we see double nickel in furrow as performing quite nicely here in the middle of the graph. And among our seed treatments, the Vellum Prime or the fluopyram seed treatment seemed to perform very well on Huntington. On high style, again, that's the orange bar for reference. For high style, I think all of the programs did positively influence yield, but those that were best included the Ridimil Gold plus Quadris and Furrow. And that seems to be a long-standing best program. I looked back in our 2019, 18, and previous years outside of the scope of this specific funding project. But we've looked at this in the past, and that's continues to be a high-performing combination, and even individually, the Ridimil Gold and the Quadris and Furrow is quite a positive one for control. It is capturing both true fungi as well as water molds. We did have Pythium and we had Fusarium in this field. And so looking at these outcomes, I know there are times that Ridimil doesn't always catch all Pythia, but it looks like we got some better control with it here. Certainly our system in this trial was overwhelmed with these pathogens, but in looking at these outcomes, again, the treatments with Ridimil Gold and or Quadris were best performing. Okay, and then final comment on the high style. With respect to the seed treatments here, we did see positive outcomes, nothing that gave us as outstanding of control as the in furrows, but one of the best seed treatments here was Saltro. And again, what Saltro is, for a reminder, is pitaflumetophin. So that's a newer adipidin fungicide from Syngenta. Pitaflumetophin is in a number of newer premixes and fungicides with Syngenta, and it's been showing to do a good job in controlling true fungal pathogens and a number of other vegetable crops as well. Right, so just to look back at past years, 1 year of data, especially when we get some funky results, it's really important to go back and look to see what has worked well in past years. What I just want to pull out from this figure again is the overall some of the differences we saw within furrow treatments compared to seed treatments. And last year we did see overwhelmingly in furrows working generally better than seed treatments, in particular for Ridimil Gold and the Ridimil Gold Quadris here. So I just want to point that out again. That seems to be the winner, at least in 2 years of our trialing. All right, so at this point, I wanted to turn the direction to discuss white mold trialing. And I think what I'll do though, before I do that, I just want to, we'll take questions at the end. I know Jed laid out the ground rules and now I'm breaking them. So I'll go ahead and I'll continue through the presentation and we'll take questions on both components of the presentation at the very end. All right, so for the white mold fungicide trial, this trial again is in its 3rd year of funding, and here we're looking at timing and selection of fungicide, and the timing has been restricted again to 30% flower and 7 days later, or 100% flower, whichever comes first. So we continue to look at treatments in that arrangement. White mold in 2020, certainly we had weather that favored white mold in some locations. It was rather spotty as it can be, as it's tied to precipitation and tied to field history. So spots where the white mold pathogen is in residence in the soil, if that overlapped with more intensive rainfall over a short amount of time, that's where we saw some of the worst white mold this summer. The pathogen does continue to be present in many or more fields than in past years, and this seems to be a growing trend overall over the past 3 decades. So as a reminder, the white mold disease cycle, it starts from the soil-borne sclerotial structure. These are sort of, they look like little raisins or mouse droppings that reside in the soil. Under the right environmental conditions, these will germinate. And typically with legumes, what we'll see happen is the sclerotia will germinate and form an apothecia, or a sort of a cup mushroom, if you will. And it's within that cup or apothecia that new spores, or ascospores, will form. And then these ascospores are discharged forcibly, and this activity is aligned with flowering of the crop. So it's aligned with that crop physiological status. An infection typically occurs through the floral parts and then spreads throughout the plant, and affects, of course, since it's the flower, will affect the pod production of the plant. The other mechanism for infection in some years, I think it was last year we saw this primarily, is direct lower stem infection by germination of the sclerotia right into the plant. So in that status of infection, you don't see these apothecia form, you don't get that ascospore infection through the flower, but you'll get a direct infection through the side of the plant. And this is important because depending on how much of a role this form of infection plays, the strategy for fungicide application could be amiss. All right. So cultural practices to limit white mold are critical. The fungicide piece is a final component, certainly an important one, but all of these other elements are essential to have in place for the fungicides to work best. So it's important to know where the pathogen is in your fields. It's important to monitor soil moisture. We do have a good understanding of the life cycle of the pathogen within soil. And so we understand how long the moisture is required and under what lighting conditions is required in order for those ascospores to form. So that is a helpful component. It's also an element of Sporecast, and I know some folks have discussed this with me. Sporecast is an app for white molds monitoring that's been developed by Dr. Damon Smith in the Department of Plant Pathology, and he has great interest in expanding this to other legumes. He's developed this tool in soybeans, and certainly the approach can be considered for other bean crops. So this is still something on our radar, and again, could be helpful. It's an app. I'm not sure if it's free now or how easily it's accessed, but if you have curiosity about this, I can certainly provide you with more information. Another important feature of white mold management is the timing of fungicide initiation. Again, when the floral infection is the main initiating point, then fungicide use prior to that peak bloom is critical. Following the cultural practices to promote drying of soil, drying of the foliage is important. Moisture on a plant will push infection, right? And so the following points all lead to reduced moisture in that crop, which include improving airflow, down and across rows, certainly avoidance of susceptible varieties. And that is often tied to the architecture of the crop, which can certainly promote continued or prolonged moisture. All right, so our study in 2020 was located at the Del Monte Plover Research Station. And that location this past summer we had quite a bit of flooding. And so we had to be selective in where we chose our plots and all of this, but we were able to still pull off a trial. But I do want to mention that for folks that were involved with that planting and that project, I thank you for that. I know it was a tough field situation, but I think we made the most of it, and we did collect information that is of use. So the planting date for this trial was the 1st of July. The two fungicide timings were the 5th of August and the 12th of August, which aligned with roughly 30 and 100% flowering. And then we collected white mold strikes or hits, as I have listed here, and yield. And then we hand harvested on September the 1st. So in this trial, we looked at some newer treatments this year, again, from guidance and suggestions from folks in industry. We looked at Propulse in 3 different rate strategies. So Propulse is fluopyram, which is vellum. So it's got that STI-HI piece, plus a trizol. It has prothioconazole, which is proline or proline. I'm not sure how you're supposed to say that, but but it has that component. And we looked at that at a 6, an 8, and then a 10.3 ounce rate. And these went out at both timings, at the 30% and the 100% flowering timings. Then we looked at Delaro. This is prothioconazole, which again is part of Propulse. It's proline, but this mixture has trifloxystrobin in it, strobilurin. And then in the second spray, we put out Propulse, which is the prothioconazole plus fluopyram. Then we had Miravis Neo. This is a relatively new registration from Syngenta. This has this flumitofen or adipidin. It has azoxystrobin or something like a quadris, and then propiconazole. So propiconazole is a different trizol than the prothioconazole, but is of this same group 3 trizol status. Then we had out Priaxor. This is fluxapyroxad, a group 7, and pyraclostrobin, group 11. And both the Miravis Neo and the Priaxor went out in just the first spray, so just a single application. And then moving down here to some of our standards, here's Topsin. This is thiophanate methyl, went out in spray 1 and 2. Then we had the Proline or the prothioconazole alone, went out in both sprays. And at the bottom of the list, Quadris, which is azoxystrobin, went out in both sprays. Missing here is Endura. In past years, we've had a whole series of those in for comparison, but I feel like I know personally in my program have a 10 years worth of those data. And we felt comfortable in comparing previous data to this as needed, but we sort of get a feel for how Endura works, and we wanted to make space for these other programs that are a bit more novel at this time. So as I mentioned, there was flooding in the field, and we had moderate white mold pressure this year. Our stats indicate that we had no significant differences by across the programs. But we do use a higher bar for our analysis. In upcoming years, I've been talking to Steve about maybe adjusting this because it's a little easier when you have the weight of your statistics to tell you what's working and what's not, right? Instead, what I find I'm doing is that we don't have these significant differences, but we do have trends, and that's what I share with you, and that's what helps to base recommendations. And so I think I might adjust that in approach in future years. Nonetheless, all the treatments did reduce white mold for both our varieties, and in this trial we had Huntington and DM88 in this year. And so Huntington is the blue bar, and the height of the bar represents yield as it has in past figures. And the gray dot represents the number of white mold strikes, right? So the higher up these dots are, the more white mold, the more disease. So you want to see these lower, and you want to see your bars taller for an ideal treatment, right? All right, and then the orange bar indicates DM88 variety. So I'm looking across here, as you see Huntington, we really didn't have any differences at all in our yield in Huntington, but we did see some differences in the white mold outcomes. And a point here toward the end, the Proline, 2 applications of Proline and 2 applications of Quadris here at the back end seem to control white mold in Huntington the best. So that was important to see. In terms of yield, though, again, no differences in that Huntington. Among the DM88 here, we generally saw higher yield overall compared to the Huntington variety. But here, the differences when compared to the non-treated control, we saw the greatest increase in yield with Propulse at the high rate. So that's the 10.3 ounce rate in 2 applications, and certainly lower disease. Here is the number of white mold strikes, and it's just over 2 per plot. So that was good to see. It certainly held back then incidence and advancement of white mold and retained pod quality and yield. The other program that really stood out was the Priaxor, single application of Priaxor. Oops, I went in the wrong direction. And so just to remind you what Priaxor is here, looking at our table, Pyraxor is the fluxapyroxad and the pyraclostrobin. And again, that was just a single application at spray 1. So that looks good. We did see a bit more in terms of disease than we did with the 2 applications of high rate of propulse, but our yield was very strong. It did a fine job with disease control in the Huntington as well. All right, so in summary here, I think I've mentioned most of these, but the 2 apps of propulse, single application of Pyraxor, and then 2 applications of, sorry, I've got a lot of Zoom content over my slide. I'm trying to discern here. 2 applications of the proline, or the proline, which is here at the end, and then the quadrus, which was at the end, that had the highest yields for DM88. So just to summarize that again for 2020. Right, in 2019, we did have a different variety there. We had 0488, but we had looked at it with Pismo last year. And what we saw there, we did see that we had good control with the proline in 2 applications last year. We had good control with Topsin and Endura as well. And some of the newer or developing biopesticides from our own bioinnovations. And so those we'll look at further in the future. But again, in 2019, we had more blossom strikes than we did in the previous years. So again, that aligns with what we did see in 2020, which were more blossom infection points rather than the lower stem, which was the case in 2018. So the 2018 summary is here. In that year, we had 3 varieties out, DMF0488, as well as Pismo and High Style. There, some of the best programs, again, included Endura, Quadrus. We saw good control with Topsin and Pismo, and some of the Marone biopesticides, again, are showing up. Best control on High Style with Topsin and Endura. So again, seeing a lot of the same fungicides with repeated best activity over the years of our trialing. I just wanted to summarize where we are in terms of registration. So in looking ahead at newer fungicides, we're seeing a lot of these premixes now coming out as the soybean and other grain crops see. We're now seeing these with registration on succulent beans. Miravis Neo we did look at this year. It did okay, but we had it in a single application. I'd like to see it at within 2 applications, and I'd like to see it maybe at a range of rates. But this is the one that it's got the pediflumetophin, the newer active ingredient, along with propiconazole and azoxystrobin. Trevopact is one we've looked at in recent years. This is it's got Topsin or thiophanate methyl in it, tebuconazole and azoxystrobin. So no brand new AIs here, but a new mix. The Revitec fungicide, as well as Provosol and Veltima. These are all newer registrations for BASF, and what makes these interesting is this mefentrifluconazole, which is a newer trizol, and I've looked at this for a number of years in potato, and it's really an outstanding fungicide for alternaria control, and we have not, aside from our Provosol treatment in this past year here. We've had it in a previous year, but we haven't looked at it extensively. It's one that I really do want to focus on for snap bean. No doubt it may be a hard fit at a price point that's appropriate, but it's just good to see where we can situate management in the future with newer active ingredients. And this one, while older triazoles may over time push a pathogen population toward resistance, this particular strategy or structure of mefentrifluconazole is one that has a unique binding conformation. And so it's one that has a little bit longer last, or a unique strategy. And so the question of cross resistance may be one that comes up further in the future as well, where we don't have such a cross, and therefore it will have positive expected activity. So with that, overall, I've just provided a run through of what we learned from 2020 and a bit from past years to help guide recommendations. And what we're finding is that the longest standing registrations are still are very good for disease control. The thiophanate methyl, boscalid, iprodione was a good to excellent treatment in our past years of trialing. The strobilurins that include Approach and Quadris provided outstanding control in our trials, as well as those from other states. And that's a component of the newer Trevo Pact. Other newer registrations, relatively newer at this point, with Fontellus and Priaxor, Proline, Propulse, are all excellent for control. Again, it's just a matter of timing and rate that that remain in some cases in question. And then an eye to the biopesticides and biologicals. These seem to come up, and over numerous years now, we're seeing very positive outcomes with these biopesticides, and so they certainly warrant further look for supporting the industry. So I want to thank you all for your partnership and for your attention here this morning.
Thanks very much, Amanda, for the great presentation. We have several questions answered, and we'll try to roll through those as quick as we can to try to stay relatively on schedule here.
Sure.
So the first question for you is, what is your confidence level in relying on products like double nickel and or other biologicals and efficacy of these products when compared back to conventional products?
Yeah, it's a very good question. And I'd say 3 years back, in our work with particularly the Bacillus species, so the double nickel is Bacillus amyloliquefasciens, and the Subtilis with Serenade, they seem more hit or miss in bean, but I would say in recent years when they've been working, they're comparable, if not better, than some of the conventionals. And we just haven't seen, at least in snap bean disease control, I haven't seen that the fluctuation like we do often see in our potato trials, where we include those biologicals routinely. So I have greater confidence in that now that that we have. In the case of Bacillus species, we've got more than 5 years of data that is positive.
Great, thank you. The next question is, do you feel there will be a variety by fungicide interaction? Some fungicides will work better on certain varieties?
Yes, yeah, and I think that's certainly demonstrated from our outcomes. There are some varieties that just seem to be more responsive to treatment. Certainly there are yield differences and other attributes that are different agronomically between varieties, but I would concur with that. I think it's a good future proposal to look at partnering specific fungicides by variety for that very reason. But it does seem that there is a differential interaction.
Great, thank you. And the next two questions are related, so I'll try to combine them for efficiency here. Sure. Have you tried combination of seed treatment and drench over the row to see if there is added seedling disease control and increased yield? And related to that, how do in-furrow treatments improve or not disease control in stands of treated seed?
Yeah, that's a good question. We didn't do those overlays in 2020. We did have some of them back in 2018. And certainly there's a combined effect, particularly when we are looking at managing two different groups of pathogens. So for example, Ridamil in furrow with a seed treatment that would be specific to Fusarium and or Rhizoctonia gives inappropriate positive interaction because you're managing different pathogens with that. But do we see synergy? And maybe I'm reading into the question a bit. Do we see synergy or increased benefit in the seed treatment plus the in furrow when they're targeting the same group of pathogens? We haven't seen that.
Okay, great, thank you. And the final question is an easy one. Were the white mold trials inoculated or natural infestations?
Well, it's an inoculated field. So the field is a white mold nursery. So it's infested and managed to promote the increase and persistence of white mold there with sunflower production between our bean rotations, but the Del Monte folks manage that as a white mold nursery. So it's inoculated, but not within the field trial season.